strings to it. That is very typical of most mushrooms. We'll have these long cells inside the stem, and that's why when you peel it apart, it's got strings. But with any Rusula, if you try to do that, it just snaps like chalk. You can't peel strings out of the stem because there are none. The cells that make up the structure of these mushrooms are called spherocysts, so they're spherical cells. The same is true for Lactarius. Lactarius and Rusula are in the order Rusulales. And we actually don't even know where that belongs in the mushroom family tree. It hasn't been determined where these are in relation to all the other mushrooms. But that's another whole nother thing. If you're in Eastern Europe, all of these are edible, with the exception of maybe 2 species, because it's a long time tradition for people to go to the forest and gather anything that they can eat, because a lot of places in Eastern Europe and it's in like Russia and Ukraine and stuff, people didn't have a lot of food, so they ate anything they could. In the United States, we don't consider most of these edible because you have to process them to make them edible. If you were to take this and cook it up at home and eat it, you would get very sick. You might not even be able to get that far, because a lot of these are either extremely bitter, like this one, or are really spicy. And so, this one's bitter and spicy. Now I'm getting the spicy. This also goes to show you that when we say poisonous, like poisonous means you would have to eat it. So if I take a bite and spit it out of any mushroom, even the most poisonous mushroom, as long as I'm not swallowing it, I'm not going to be poisoning myself. But Rusula, these can all be, with the exception of 2, and it's actually the ones that turn black. So if you find a black Rusula, you definitely can't eat that. But the rest of these can be detoxified and made into pickles and other things. I recommend doing a lot of research before doing that. I personally don't do it because I'm not a huge fan of them. The texture is often mealy and gross and chalky. There's a reason why most Rusula aren't considered amazing edibles. There are a handful, though, like the shrimp Rusula that comes out earlier in the year. It's large, it's substantial, and it has like an odor of shrimp and a taste of shrimp. And it's a really delicious mushroom. But for the most part, few people eat Rusula in the United States.
What are the round ones?
The round ones. So these are probably. This is a young puffball. So this one right here, Lycoperdon umbrinum. When it's a baby, it's got this white flesh. And they're actually edible, but when it grows up, it becomes, becomes a puffball. So this creamy white stuff on the inside, which is called the gleba, would mature and just turn into pure spores. This one is a young Amanita. Amanita are, I couldn't tell you which species, but Amanita are some of the best tasting mushrooms, but also the most deadly mushrooms. The death cap, Amanita phalloides, has killed more people in the world than any other mushroom in history. And it is in California. You are very unlikely to find it here in Salt Point State Park. So you don't have too much to worry about with that specific one. But in the Bay Area, under Coast Live Oak, you'll find Amanita phalloides, the death cap. You'll find that during the fall. Also in the spring. It hasn't really figured out a temporality yet because it's invasive. It came to the United States on a cork oak in the 1980s, and I think it came into a nursery in Mendocino and spread out from there. And since the 1980s, it has spread across the entire United States. It is associated with dozens of different species of trees. It's actually a really insane, from a biogeographical perspective, because usually invasiveness doesn't happen on that scale. And it can be mistaken. Folks from Southeast Asia who are used to the paddy straw mushroom, which is a Volvarella species, it looks a lot like Amanita phalloides. And so during a time when a lot of people from Vietnam were moving to the United States, it looked just like the one they were finding at home, and there was a rash of deaths in the early 2000s of people consuming them. So that one, Amanita, that whole genus is something you have to be definitely an expert to start digging into. There's a few edible species though, none of which I see on this table, but one of them grows. Yeah, I don't see. This is the only amanita I see right here.
There's one over here.
Oh, there's one.
A bigger one.
Okay, that one's kind of rotten. I don't even want to touch it. Yeah.
Like a hamburger bun.
Yeah. Yeah. Before you handle that, just be aware that your hands are going to smell like fish.
Is that
It's
a,
gross.
is that a,
That is an amanita. Yeah, that's Here's a good example of an amanita. So usually amanitas have some sort of feature on the base of the stem, and that is called the vulva, and it's from the way that the mushroom develops. So when the mushroom develops, it's under the ground. It's even more compact than this. It's like an egg, and as it grows, it pushes out of the base of the egg, and then there's a covering over the top, and that breaks up into these little dots on top. And then the covering that goes over the gills ends up becoming a ring around the stipe. So a lot of them do have a ring. This one has remnants of a ring. It's kind of old. It's been handled a lot, so there's no like really beautiful crisp ring around there, but you can see the remnants of it. So this is probably Amanita pantheranoides or Ameripanthera, and I don't remember how to tell the difference between them. This one contains ibotenic acid, which is a toxin, so you wouldn't want to eat these. They also contain mucimol, which is in Amanita muscaria, the big, like, when you think of like a toadstool mushroom with like the red top and the dots on it, that's what you're thinking of as Amanita muscaria. Mucimol is a intoxicant, and some people would call it a hallucinogen. This one does contain small amounts of that, but it does have ibotenic acid, which is a neurotoxin.
before you get that really fun reaction?
Nobody I've heard of who tried it described it as fun. It's one of those things where like you take it and then like you sweat and feel terrible, and then you wake up somewhere and you don't know how you got there. You might have clothes on, you might not. Might be like, in the supermarket pushing around the thing and being like, oh, why am I naked in the supermarket?
Amanita very rarely kills people.
Oh, okay.
Yeah. So the it is interesting. I'll go a little bit into detail. So we don't have any Amanita muscaria on the table to show off. They're really beautiful mushrooms, and they're very common out here during the late fall and winter. They kind of co-occur with porcini. So if you see Amanita muscaria, you know that those are the same areas where you'll find porcini. So, but Amanita muscaria is a worldwide species, and it's been used for its psychoactive properties by a lot of different cultures. In Scandinavia, there's this sort of legend that people would drink reindeer urine, and that's because the reindeer would eat the Amanita muscaria mushrooms. The poisonous thing in there called ibotenic acid, when it's metabolized by someone's body, it turns into the psychoactive one. So the reindeer would eat it and then convert the poison into the psychoactive component. And that's true with humans as well. There's several cultures where people would have the shaman consume the Amanita muscaria mushroom. Their body would turn the ibotenic acid into mucimol, and then they would urinate and pass it around. We don't do that anymore. So,
Except in Silicon Valley.
yeah, maybe in Silicon Valley. I don't know. I don't live down there. I don't know how you all do down there. But it has a whole host of compounds in it, and a lot of those are actually being evaluated as medicine. So a lot of people do make tinctures of Amanita muscaria and will take very small amounts of it to the point where the toxin doesn't have any effect, nor does the hallucinogenic compound. But people claim that in taking small doses of it, they have relief from chronic nerve pain and other disorders. People can who have trouble sleeping can take tinctures of Amanita. So there's a lot of layers to this. That's all about all I have for Amanitas, except that the most delicious mushroom, in my opinion, in California is Amanita villosa, and it's fruiting right now under Coast Live Oak. one of the most deadly mushrooms in the world, Amanita ocreata, is also fruiting under Coast Live Oak. So it's something to aspire to. When you can tell those two apart confidently, you will have unlocked the best tasting mushroom in California. But you don't want to make the mistake. So it contains a toxin called amanitin, and what it does is it stops RNA polymerase II. And RNA polymerase makes RNA, and RNA turns into proteins. So if you can't make RNA, you can't make proteins. And if you can't make proteins, basically all functions of your body cease. And it's a slow process. So you eat it, gets absorbed into your GI tract, and basically all the cells in your GI tract die. So you get really sick, you throw up, you have diarrhea, but then you feel fine. It absorbs into your liver at that point, and you don't feel when your liver is being destroyed. Those cells don't make you vomit or anything. But in a few days, your skin starts to turn yellow, the whites of your eyes turn yellow because your liver is completely damaged, and then the toxin moves on to other parts of your body. So you really don't want to make that mistake. It is curable, but a liver transplant is about $1 million with installation. So
I'm stealing the Dermosophy smithii. These are a dye mushroom. So I'm taking these for our camp next January, so I'm stealing them.
yeah, that's the one that I put the KOH on, and it turned one of them. Let's see, there was a little one here.
Yeah.
Well, one of them, yeah, that one you put the potassium hydroxide. It looks really.
We're gonna just sign that out. We want to see it.
No.
Yeah.
It does make a really beautiful color. They're almost the same color, and it's color fast dye. Yeah. So.
Can you trade with me?
Dermosophy smithii.
Who does dyeing with Dyer's polypore?
Dorothy Baby and.
What color does this make?
A golden yellow
Like gold
color.
yellow.
And can you use it when it's this old?
You can
Not
totally.
really. Like,
Oh, okay.
When it's old and dried up, it totally works.
Wow. I was always under the impression that you needed it fresh.
I like
That's-
them fresher.
That's cool.
all right. Thank you. Thanks everybody. Good to see you, everybody out here today.
Something. Let's do what that is.
Yeah, that's also a Cortinarius. I think, or Ninosabe. Let's. I can't tell by smell. Let's try the KOH. If it's a Cortinarius, it's probably in Dermosophy, and it'll turn a color. Let's see. Yeah, it's Cortinarius in section Dermosophy. A really deep red, like really like the reddest red wine. This is a 5% solution of potassium hydroxide. It's what people use to make soap. So you wouldn't want to like get it all over your skin because it would turn your skin into soap. Exactly. At 5%, it's not that strong. It's probably not going to hurt you, but don't use it as eye drops. I've definitely had it spill in my pocket and been fine. But yeah, as far as diversity goes, this is not a great time to be hunting mushrooms in California. Typically, this time of year is the heart of mushroom season, but we had a couple of things kind of converge on this season to change things. One was the rain that we got in September. We got pretty significant rains in September, and the mushrooms are waiting for that. Once they get that big soaking, they're like, okay, season's on. So they got started, like 6 weeks earlier than they normally get started, and that got everything going early. Then we had that big storm in January, and normally rain is really good for mushrooms. But mushrooms breathe air just like we do, and when there's so much rain that everything is completely inundated with water, they have a hard time growing because they like everything's just soaked. And these really heavy storms that knock trees over and flood places and move the debris around, mushrooms don't like that very much. They're living in the leaf litter, they're living in the debris, and so when that just gets torn up by these storms, they kind of lose their vigor. Then it's also been really cold. So cold temperatures will slow down mushroom fruiting, and if it's below freezing, you're not going to really get that many mushrooms at all. What you typically see this time of year is going to be stuff, the winter trio, which is the hedgehog, the black trumpet, and the yellowfoot chanterelle, all right over here. So this is the winter trio. And this typically goes from December all the way into March. So these are probably not going to last until March, but you'll probably find them for the next few weeks at least. Jackson Demonstration State Forest in Mendocino has some great habitat for these. They're all in Cantharellacea, all in the same family, even though they all look super different. The nice thing about this, and especially the hedgehog, is that it's really hard to pick something that is a look-alike. Like, there's not really that many mushrooms that look anything like that. At worst, I don't know what this is. I think is this Bulgaria? I don't know. Is this Bulgaria, do you think?
No, that's Exidia?
Oh, Exidia, yeah. Yeah, I mean, and this doesn't really look the same as this, you know, maybe superficially, but this obviously has a stem. It has this trumpet that opens up. These have teeth. It's pretty hard to mistake the teeth. There are other things that have teeth out there, but they would be really hard, and you would know pretty quickly that they weren't edible because you wouldn't be able to actually eat them. So.
Do all of the yellow feet have the holes in the top?
Not universally. So this young one here does not have a hole in the top, but if you were to kind of peel it open, oh, it's not even hollow yet. When they grow up, they're always going to have a hollow stem. But the biggest way to tell these apart is just the fact that they have ridges instead of gills.
How much of the stem would you leave on if you cooked them?
All of it. Oh, the whole mushroom's edible. That's in most cases. You rarely have to cut the stems off. The other thing that we would typically only just start seeing right now would be the waxy caps, but we're already almost through all the waxy caps. These, this is in the family Hygropheraceae, and it includes the most colorful, amazing looking mushrooms that you'll ever see. Like this big bright red one, which is Hygrocebae punacea. This is Hygrocebae flavescens group, and this is Hygrocebae marchii. And these names, I'm using them with very little confidence because a lot of these species were described in Europe. In Europe, you would find these growing in grassy areas. But for some reason, here in California, they grow in like redwood forests and in totally different habitat. We really don't know much about the ecology, other than that they start to show up this time of year here in California, and they always grow with a certain assortment of mushrooms that we call chegs. And it's short for Clovariaceae, Hygropheraceae, Entelomataceae, and Geoglossaceae. And I only see one example of that group here, and that's the H, the Hygropheraceae. They're really brightly colored, not because they're poisonous, but as far as we know, we don't know if they're edible. There's conflicting reports. If you look in Mushrooms of the Redwood Coast, which is the best book for eating mushrooms in California,
I beg to differ, sir.
oh, you have a contender.
Well, I do.
What?
For the beginner, it's Mushrooms Demystified is the best single work, but
Fair, but
it's open to debate.
yeah.
But what I would like to interject, Hart mentioned, they saw the mushrooms in Europe, they came over here, and they said, oh, it's the same thing. Well, it's a different continent, it's a different habitat, it's a different soil type, on and on. And the more they get studied, they find that no, it's not the same species as the one that looks like it in Europe. And so you're going to see constantly new names coming up, because science is a self-correcting view of the universe, and we keep learning more stuff. So like your classic sulfur shelf, the chicken of the woods, evidently it's about 7 different species in North America, and it's not the same species as you find in Europe. So you're going to be faced with ever-changing names, but that's a little bit of the background for how some of these got named.
Yeah, very true. Yeah, most of the people that study mushrooms in the United States were European mycologists. So they applied the names that they had from Europe. Now the pendulum's actually swinging the other way. So there's been this big push to do DNA barcoding on mushrooms. And DNA barcoding is where you just look at a short piece of DNA, like 600 base pairs long. The whole genome of a mushroom is 50 million bases. So you're just looking at a little short sequence that changes over time at a predictable rate. So you can kind of say, oh, if it has this barcode, then it must be this species. If it's a little bit different, then it must be a different species. And we've done a lot of work using that technique. The pendulum is now swinging the other way. Now we're starting to look at whole genomes. So we're not just looking at a little 600 base pair fingerprint, we're looking at the whole 50 million base pairs. And what we're starting to see is that we're actually wrong about using the term species. Mushrooms may fall outside of the traditional category of a species, which would be, there's a couple of different concepts. There's the biological species concept, which is like, there are two different species if they can't mate with each other. But that kind of falls apart when you have like mules. So, so there's a couple of different things. Morphological, oh, it looks different. Ecological, it has a different ecology. And those are kind of traditional ways we've been applying to describe mushroom species. By looking at whole genomes, there's a recent paper that was published about porcini, and they did 168 whole genomes of porcini across the entire world. And they're able to see it grouped into 6 different populations. These mushrooms look fairly similar, but there are some differences that you can tell. They do taste different, they do have different habitats, and they were all described as different species. But they were actually grouped back together into the same species in this paper, because what they were able to see is that all of them shared the same set of genes for about 40% of the genome. The other 60% of the genome were genes that they had acquired. So I'm not talking about genes that evolved. It's not like, you know, this mushroom evolved to have teeth because that was a better way of distributing spores. It was more like the mushroom moved into a certain ecology and somewhere got a whole bunch of genes from the environment through what's called horizontal gene transfer. So vertical gene transfer is pretty common. It's like, I have a child, they have the same color eyes that I had. Horizontal gene transfer would be like, I go up and shake your hand and give you my eye color. So something like that has been happening with fungi. So we might actually swing back in the direction where we start grouping things back together and we apply a different species concept, which is the same one that we apply to bacteria. And it involves the idea of what's called a pangenome, where you have a whole lot of different genes that can be within a species, but each individual population has a different subset of those genes. You will always have one core genome that is the same throughout all of them, and then you'll have accessory genes. So my theory is that since porcini are mycorrhizal mushrooms, they associate with trees, when they become established in that environment and they start to connect with the trees, that there is some mechanism where they receive the genes to be able to interact with that tree. We don't really know the mechanism behind that. We do know that horizontal gene transfer happens in bacteria really often, where they will just exchange genetic material all the time, and that's kind of the same thing is happening with fungi. We just don't know what's going on. I mean, the important point is that when you find mushrooms in the forest, you're looking at their reproductive structure. If you looked at a person and tried to understand their ecology just by looking at their reproductive structure, you wouldn't get very far. There's a whole organism underneath the soil. This is just this little thing sticking out. There's this whole organism, and it interacts with countless organisms. If you take 1 gram of soil and sequence all the DNA in there, you'll find thousands of different bacteria, fungi, arthropods. So there's a whole ecology that you just can't see. It's called the rhizosphere. It's what's happening around the roots of the trees. And hopefully by doing these whole genome analysis, we'll start to get a better idea of what's going on. But that was the tangent that I went on.
Science!
Yeah, and one of the things that I'm observing is